the last class of microbiology today. Today we are going to study the family Retroviridae that you already know. So we are here. In this picture, we are here. Retroviruses. Retroviruses are single-stranded RNA viruses with positive sense. And you already know them because they belong to the Baltimore class 6, and they are special because they can do something strange. They can perform reverse transcription. They can make DNA using RNA as a template. Okay. The family Retroviridae contains many different viruses. Important for humans are only two. The HTLV, human T leukemia virus, and the HIV. However, the first one, we don't have time to study. So I hope that in the future you study the disease that they cause, because in this class we have no time. In this class we are going to focus only in HIV. So we have HTLV and we have HIV. We focus in HIV. HIV viruses, human immunodeficiency virus, are two types, HIV-1 and HIV-2. The most common is HIV-1. The other one, HIV-2, is only found in West Africa. And they are not exactly the same. The disease that they cause is the same, or almost the same, but not exactly the same. In this lecture, we are going to focus on HIV-1. Retroviruses were discovered 40 years ago. The first retrovirus that was discovered was the HTLV by Robert Gallo. This virus, HTLV, is oncogenic. 4 years later, in 1984, François Barré-Sinoussi and Luc Montagnier discovered HIV at the Pasteur Institute in Paris. And they discovered that this virus is the cause of a terrible disease that is called AIDS. You already know that they received the Nobel Prize of Medicine in 1908, as you already know. Okay, so for all retroviruses, all retroviruses, the nucleic acid is positive single-stranded RNA. They can perform reverse transcription. Retroviruses contain two copies, identical copies, of the genome in each virion. So each virion contains two copies of RNA, as you will see later. About the structure of the virion, the capsid, and the envelope, I will tell you later when we will talk about HIV. So look, this is the structure of all retroviruses, not only HIV. Two copies of RNA, the capsid, the envelope with glycoproteins, and some enzymes are also found inside. But I will explain you later in more detail. So look, what about the viral cycle? In summary, because later I will explain you with more detail. In summary, sorry, wait a moment. We are here. Can you see the whiteboard? No. Wait a moment. Okay. So look, what is reverse transcription? What is reverse transcription? These viruses, so this is reverse transcription for all retroviruses, all, not only HIV. So HTLV, HIV, and also others. What's reverse transcription? These viruses, the nucleic acid is positive single-stranded RNA. Okay, this is the original nucleic acid. So we can make this drawing. This is single-stranded RNA. Okay. The first step of reverse transcription is to make single-stranded DNA. So using positive single-stranded RNA as a template, these viruses perform the complementary single-stranded DNA. This is the first step of reverse transcription. Actually, that's the meaning of reverse transcription, to make DNA from RNA. So this is single-stranded DNA. This is single-stranded DNA. And the second step of reverse transcription is to make double-stranded DNA. This is the second step. So after the second step, retroviruses have double-stranded DNA. This double-stranded DNA of retroviruses is called provirus. In Latin language, pro means before. Provirus, double-stranded. So look, reverse transcription has two steps. The first one, to make single-stranded DNA from RNA. The second one, to make double-stranded DNA. Both steps are performed by the same enzyme, the reverse transcriptase enzyme. Both steps. So look, this enzyme, the reverse transcriptase enzyme, has three functions. The first one, to make single-stranded DNA from RNA. First function. Second one, to destroy, to remove the original RNA. Second one. And the third one, to make double-stranded DNA. So this enzyme, the reverse transcriptase, has three functions. This is the process of reverse transcription, okay? So finally, from positive single-stranded RNA, these viruses make the provirus. The provirus is the double-stranded DNA. Okay, and what do they do later with the provirus? The double-stranded DNA, this is the provirus, is then integrated inside the host cell genome. So after that, the viral DNA, so the provirus, is integrated inside the host cell genome. This process is called integration, and this is made by another enzyme. This enzyme is called integrase, that I will explain you later. So this is common for all retroviruses, not only for HIV. Okay, so we go back to the presentation. So this is the same that I explained you here in this picture. Later, we will watch a video related only with HIV. As I told you, some retroviruses are oncogenic, not HIV. HIV is not oncogenic. Other are oncogenic. For example, HTLV. So this is the same that I explained you. So look, the last step in the flow of genetic information is to make proteins. How can they do that? The provirus, so the double-stranded DNA, is integrated into the host cell genome. So how can they make proteins? The first step to make proteins is transcription. And the second one is translation. Transcription is made by the cellular RNA polymerase II. You already know from cell biology and from biochemistry that this enzyme belongs to the human cells. This is the enzyme that human eukaryotic cells use for transcription to make messenger RNA. Because the double-stranded DNA from the virus, the provirus, is integrated into the host cell genome, the virus can use the cellular enzyme for transcription. You will see later in the video. Okay, so now we are going to focus only in one. We are going to focus in the human immunodeficiency virus, in HIV. 40 years ago, something very strange happened. In the year 1981, you don't know because you are very young, but I remember. At that time, some people in the US, very young people, went to the hospitals because they were suffering very strange opportunistic infections, and nobody knew why. These opportunistic infections were usually pneumocystis jirovecii pneumonia. This is for next year because this is a fungus, or Kaposi sarcoma that you already know from the class about herpes viruses. These people were very young and they were suffering severe opportunistic diseases, and these patients were apparently not immunocompromised and nobody knew why. Some years later, actually 3 years later, Françoise Barré-Sinoussi and Luc Montagnier in the Pasteur Institute in France, they discovered that HIV infection was the cause of this severe immunodeficiency. This severe immunodeficiency was called AIDS. Acquired immunodeficiency syndrome was first recognized in the US. This is the picture of Kaposi sarcoma that you already know because you studied in the class of herpes viruses. Okay, so we have two types of HIV, HIV-1 and HIV-2. The most common in the world is HIV-1. This class is about HIV-1. HIV-1 can be divided into 4 groups: Group M, Group O, Group N, and Group P. The most common in the world that you must remember, this is one of the questions that you have in the quiz, is the Group M. Around 90% of HIV-1 infections are Group M. So Group M is the pandemic form of HIV, the most important. Group M may be further subdivided into 9 subtypes that you don't have to remember now. Just remember, HIV-1 Group M is the most common in the world. This is the pandemic form of HIV. HIV-2 is very similar. The disease is also very similar, but not exactly the same. HIV-2 is mostly found in West Africa, in the countries of Guinea-Bissau and Senegal. But because this virus is not so common, the rest of the class is related only to HIV-1. What about the epidemiology? Currently, this is a pandemic. Currently, HIV infection is the most important public health challenge in the last 30 years. Currently, between 30 to 44 million people are living with HIV. The prevalence in the world is around 0.8%. Around 10% of them are children, younger than 15 years. In the last 40 years, more than 25 million people have died already. And importantly, and very sad, around 90% of the new infections occur in the poor countries of the world, mostly in sub-Saharan Africa. We are here. These are the last data provided by the World Health Organization. In Southeast Asia, we are here. We find around 4 million people infected with HIV. I like this map very much because in this map, we can notice, we can understand the importance of the problem related with the size of the country. So look, the problem of HIV infection is mostly found in the poorest countries of the earth. So the problem of HIV is mostly related with sub-Saharan Africa here. You can find this map in this website that I like very much. And you can find these maps related with many different diseases. They are very interesting. However, something good has happened in the last 10 years. In the last 10 years, the number of new infections have decreased 16%. The number of deaths have decreased 33%. And what about Vietnam? These are the last data provided by the World Health Organization that you already have. In Vietnam, currently we have around 230,000 people living with HIV. Children, just 5,000. So the prevalence in Vietnam is 0.3%. So this is much lower than the prevalence in the world. And this is much lower than your neighbors, Thailand. The prevalence in Thailand is much higher. It's almost 1%. And currently in Vietnam, around 65% of infected people are receiving treatment. And we have improved in the last years. However, we need to treat all of them. So we still have to improve this. The good news is that almost all children, around 92% of infected children, are already receiving antiviral treatment against HIV. And that's very good. Vietnam has improved very much in the last 10 years. In the last 10 years, the deaths in Vietnam because of HIV have decreased 45%. This is very good. And the number of new infections in Vietnam in the last 10 years have decreased about 64%. Okay, and we start now with some things more related with microbiology. The most important thing that you must remember, the transmission of HIV. Currently, we know perfectly how HIV is transmitted. We know it perfectly. The most important route of transmission is sexual contact. Sexual transmission is the most important route of transmission in the world, the most common. Sexual relationships from men with women. So heterosexual relations, the most common. Parenteral transmission, so through the blood, is also possible. For example, injection or transfusion of contaminated blood, or sharing needles, contaminated needles. This is a common source of transmission in people who inject drugs. Vertical transmission is also possible, so mother to child. Vertical transmission is possible during pregnancy, at birth, and during breastfeeding. Around 15 to 30% of mothers infected can transmit HIV to the child. Most of them, around 75% of these transmissions, occur during late pregnancy, so during the third trimester, or during delivery. So remember, the transmission of HIV is possible through sexual transmission, through parenteral transmission, and vertical transmission. So look, you already know 3 viruses that are transmitted by these mechanisms: HIV, hepatitis B, and hepatitis C. Actually, we can find some people infected by 3 viruses at the same time. They are usually heroin drug users because the mechanism of transmission is through the parental route. So look, when we compare HIV with other diseases, for example, influenza or the new coronavirus, currently we know that HIV cannot be transmitted so easy as influenza. Influenza virus and coronaviruses are transmitted very easily, but not HIV. So we know perfectly how to protect ourselves from transmission, because we know that transmission is possible through sexual contact, parental contact, or vertical transmission. So it's much easier to protect ourselves against HIV than to protect ourselves against influenza, for example. Okay, and we start now with the real microbiology. This is the structure of HIV. Before this picture, I want to explain you something in the table. So look, the nucleic acid, so the genome of HIV contains many genes. 3 of them are the most important. The most important genes found in HIV are these 3 genes. I write again so you can follow the explanation. The GAG gene, the POL gene, and the ENV gene. These 3 genes are the most important in HIV. And what do they codify? What proteins do they codify? What about this one? The GAG, the GAG gene, codifies proteins for the capsid, for the core. But not only, also other proteins. The POL gene codifies, encodes enzymes, 3 enzymes that I will tell you later. And the ENV gene encodes proteins for the envelope, hence the name. So look, the most important protein from the capsid that you must remember is the protein 24. The P24 is one of the proteins of the capsid that you must remember for later. But not only the P24, also other proteins from the capsid, the P6, the P7, and others. But these are not so important for you, so you can forget. Just remember the P24. What about the enzymes? HIV virus contains 3 enzymes that you must remember. The first one, the reverse transcriptase, that you already know. The second one, the integrase, that you already know. And the final one, the protease, that you will understand later. These 3 enzymes are codified, are encoded in this gene, in the POL gene. And these enzymes are very important in the viral cycle of HIV, as you will see later in the video. And finally, about the envelope proteins, you must remember the glycoprotein 120 and the glycoprotein 41. This glycoprotein, the 120, is the glycoprotein that the virus uses for binding host cell receptors. So you must remember these proteins. These proteins are the most important for HIV virus. So this is the structure of the virion. Nucleic acid, remember, 2 copies of the nucleic acid, 1 and 2. Positive single-stranded RNA. Both copies are identical. They have the same genetic information. This is the capsid. The capsid is made of proteins. The most important protein of the capsid that you must remember is the P24. And this is the envelope. The envelope contains 2 glycoproteins. This one with green color, this is a transmembrane protein. This protein is included inside the envelope. This is the GB41. And this one with purple color, this is the GP120. This is the protein that the virus uses for binding host cell receptors. Okay, so what about the viral cycle? So we are going to watch the video because this video is very good and we can understand perfectly the viral cycle. Wait a moment, because I have to find the video. So the video is here. Wait a moment. Okay, so can you see the video? Yes. So look, so this is the virion, okay? This is the HIV virion. And these spikes with yellow color, this is the GP120. This is the glycoprotein that the virus uses for binding host cell receptors. So look. So this is the capsid with 2 copies of the nucleic acid, identical copies of the RNA. And this is an enzyme. This is the reverse transcriptase enzyme. And this yellow is another enzyme. This is the protease that you will understand later. So look, first step. The first step is binding to the host cell. So the first step is attachment. And look, I will explain you later the host cells. So look, this protein, this is the glycoprotein 120, the viral glycoprotein for binding receptors. This is the host cell. This red protein here and here, this is the host cell receptor. The host cell receptor for HIV is the protein CD4 that you already know from the second class. The protein CD4 here with red color is the protein that the virus uses for binding, is the protein that belongs to the host cell that binds the glycoprotein 120. However, look here, with blue color here, I show you another protein. This protein is called co-receptor. So look, because this is very important. HIV virus needs 2 proteins for binding. So the glycoprotein 120 binds both the receptor and the co-receptor. The receptor is the protein CD4 that you already know. The co-receptor is one in blue color. I will explain you later, and I will explain you later the meaning of the co-receptor. So this is the binding to both, to receptor and co-receptor. First step. Second step, fusion of membranes. The viral envelope fuses with the host cell membrane. Fusion. So the capsid enters inside the host cell. Next step, uncoating. You remember from the first class of virology, uncoating, removing the capsomers to release the nucleic acid. This is uncoating. So now the nucleic acid, both copies of the RNA are free in the cytoplasm of the host cell. And look what happens now. Reverse transcription starts right now. Look the steps. This is this yellow nucleic acid. This is the original positive single-stranded RNA. This blue protein, white blue protein, this is the reverse transcriptase enzyme. Reverse transcription is going to start right now. Look. So first step of reverse transcription. The RNA positive is transcribed into single-stranded DNA. This is the first step. Yellow color, RNA. Purple color, DNA. So look, during this step, two things are happening. The first one, the synthesis of the single-stranded DNA. First thing. Second thing, the RNA, the original RNA is destroyed, is removed. Both functions are performed by the same enzyme, by the reverse transcriptase enzyme. This is the first step. Second step. The second step is also performed by the reverse transcriptase enzyme. During the second step, the single-stranded DNA becomes double-stranded DNA. Look, this is double-stranded DNA. This is the provirus that I told you before. And look what happens right now. Another enzyme. This is the integrase enzyme. This is a viral enzyme that I told you before. This enzyme is going to integrate the provirus inside the host cell genome. So it goes to the nucleus, it enters inside the nucleus, and this is the double-stranded DNA of the host cell. And this is the provirus. So look, the integrase, the enzyme integrase, integrates the provirus inside the host cell genome randomly, wherever, here or here, wherever it finds. No, well, actually, I show you in the video only one copy, but this happens many times. In each host cell, between 1 to 20 different copies of the provirus are found. Anyway, so currently, the provirus has been inserted, included inside the host cell genome. So now it's very easy to make proteins. Now, the cell enzymes, the RNA polymerase II, can perform transcription and can make messenger RNA. This is the cellular RNA polymerase II. Transcription. This is messenger RNA that will be translated into different proteins for the virus. For example, this messenger RNA is going to be translated into the glycoprotein 120. This is messenger RNA from the virus. This is the glycoprotein 120 that will be taken from the envelope later. Another messenger. This is another messenger RNA from the virus. Look, another protein is going to be translated. This protein, this is a polyprotein or multiprotein. That means this protein contains different subunits. This polyprotein, this multiprotein contains different proteins that will be cut, cleaved later. So this polyprotein goes to the new virion that is going to be released right now. So this is the new virion. The new virion contains the glycoprotein 120 at the envelope and many polyproteins inside. And look what happens now inside the virion. These are the polyproteins. These polyproteins are cut, cleaved right now by this enzyme. This is another viral enzyme. This is the protease. So remember, HIV virus contains 3 enzymes: reverse transcriptase, integrase, and protease. The function of this enzyme, the protease, is to cut the polyproteins. So this is the new virion. Now assembly of the capsid, and the new virion is released outside. So this is the viral cycle of HIV. We go back to the presentation. This is the same, but the video is very good. If you watch the video, you can understand all the steps of the HIV viral cycle. So what about the damage of HIV? What about the pathogenesis? So what's the problem? The problem is that the target host cells infected by HIV belong to the immune system. So the damage means a progressive, slowly loss of cellular immunity. After many years, very slowly, HIV-1 infection progressively kills T helper cells after many years. So after many years, when most T helper cells are destroyed, the patients may suffer a very severe immunodeficiency. This is called acquired immunodeficiency syndrome. So the patient may suffer many different opportunistic diseases. So what about the target cells? Look, the most important target host cells are T lymphocytes CD4 positive. This is the most important target host cell. These T lymphocytes are called T helper cells. as you already remember from the second class. Macrophages, some macrophages are also infected by HIV. Some dendritic cells, remember dendritic cells from the second class, and some cells of the brain, microglial cells. Actually, microglial cells are macrophages that are found in the brain. You will study them in histology. So look, all target host cells infected by HIV-1 belong to the immune system, all of them. The most important, T helper cells. So this is the problem of HIV-1. HIV attacks at the very heart of the immune system, because T helper cells and macrophages are very important for both humoral and cellular immune responses. HIV attacks the very heart of the immune system. I told you 2 weeks ago that when a virus attacks cells of the immune system, we always have serious problems. So look, the most important host cell destroyed by HIV are T helper cells. T cells CD4 positive. T helper cells may be destroyed by 2 different mechanisms. Directly by HIV. So the HIV virus destroys the T helper cell. However, T helper cells may be also destroyed indirectly by the immune system. For example, why? Because T helper cells become targets of the immune system because they are infected by the virus. So other cells from the immune system are going to kill the infected T helper cells. So this is another civil war inside the immune system. Healthy T helper cells and T cells CD8 positive are going to destroy the infected T helper cells. T helper cells are going to be destroyed by apoptosis. You already know apoptosis from cell biology. Apoptosis is like suicide. Some T helper cells kill themselves because some proteins produced by the HIV are toxic. But we are not going to study this in detail, okay? Just remember, T helper cells are destroyed by the virus itself or indirectly by the immune system and by other mechanisms. The immune system reacts to fight against HIV. The immune system develops antibodies, so humoral response. Many different antibodies are produced against HIV. Antibodies against glycoprotein 120, antibodies against the glycoprotein 41. And these antibodies are neutralizing. These antibodies are very good. So what's the problem? The problem is that HIV quickly escapes because of many mutations. So these antibodies can work at the beginning, but not later, because the virus is changing very quickly because of mutations that I will tell you later. So humoral immune response, antibodies, and cellular immune response, T cells CD4 positive and T cells CD8 positive. All of them will fight against infected T helper cells. So look, we are going to study in detail the first step. The first step of the viral infection is attachment. This is the first step, when the virus binds the host cell. So look, the viral protein that the HIV virus uses for attachment is the glycoprotein 120. This glycoprotein that you must remember perfectly is found in the envelope. What about the host cell proteins that bind the viral protein? The host cell receptor is the protein CD4. So all cells that contain the CD4 protein may be infected by HIV virus. However, as I told you during the video, the HIV also needs co-receptors. And two different types of co-receptors may bind HIV. These co-receptors are also proteins, and we have two. The first one, CCR5, and the second one is CXCR4. These are proteins that you don't know yet because you don't know immunology, but you will study in detail next year. These proteins are receptors for cytokines that are found in cells from the immune system. So CCR5 and CXCR4, co-receptors. So HIV needs two proteins for binding, the CD4 and the CCR5 or the CXCR4, the co-receptor. So look, this co-receptor is found mostly in macrophages, but also, but not so important, in T helper cells. So in T lymphocytes, CD4 positive. However, this co-receptor, the CXCR4, is only found in T helper cells, not in macrophages. So look, we can differentiate, I'm going to delete this, sorry. So we have more space. We can differentiate two different HIV, well, HIV1, HIV strains. Two types. The first one, we call them M-tropic strains. And what is this? These M-tropic strains are HIV strains that bind the CCR5 receptor. And why do we call them M-tropic? Because they bind macrophages. Because this co-receptor is mostly found in macrophages. M-tropic strains mostly bind macrophages, hence the name M, because they bind the CCR5 co-receptor, also to T helper cells, but less. And the second type, the second HIV strains that we can find inside a patient are the T-tropic strains. The T-tropic strains bind to the other co-receptor, CXCR4. And why do we call them T-tropic? Because they mostly bind T helper cells, because this co-receptor is found in T helper cells. So M-tropic strains and T-tropic, and they are not exactly the same. These HIV strains are different. Which one is the worst? The T-tropic. The T-tropic strain is the most dangerous, the most virulent. The T-tropic strain can destroy T helper cells more efficiently. So the T-tropic strains are the most virulent. What about the M-tropic strains? The M-tropic strains are not so virulent. However, the M-tropic strains are the most contagious. So they are more easily transmitted from patient to patient. When a patient is infected at the beginning, the most common strains that we find in the patient are the M-tropic strains. So the disease can be transmitted more easily at the beginning because these strains are more contagious. When a patient is infected, a patient is usually infected by an M-tropic strain. At the beginning, when a patient is infected, M-tropic strains are the most commonly found. However, these M-tropic strains mutate later because the virus mutates very easily. So there are many mutations. Many mutations are found in the M gene. Remember, the M gene encodes the glycoproteins. So, after these mutations, new strains appear. And these new strains start binding the other co-receptor, the CXCR4 co-receptor. So at the end of the disease, the most commonly found, the strains most commonly found are the T-tropic. At the beginning, when the patient is infected, M-tropic strains. At the end, T-tropic strains. And the T-tropic strains are the most virulent because these T-tropic strains can destroy the T helper cells more efficiently. M-tropic and T-tropic. In the middle, we have another one. In the middle, we have strains that can bind both co-receptors, this one and this one. So this is very complicated. The process, the pathogenesis is very, very complicated. I only show you here an introduction. So just remember the M-tropic strains and the T-tropic strains. The M-tropic appear at the beginning. They bind the CCR5 receptor. They mostly infect macrophages. They are very contagious, but not so virulent. After many mutations, new strains appear, T-tropic. They bind mostly T cells because they bind to the other co-receptor. They are the most virulent, but the transmission of these strains is not so easy. So the transmission of HIV is easier at the beginning because the patient is infected by M-tropic strains. Okay, so we go back to the presentation. So this is the same. Here I explain you receptor, co-receptors, and both strains. So the problem of HIV is that the reverse transcriptases are not very good, and they make many mistakes during replication. This problem also appears with, I think, with all RNA viruses, all. So mutations in RNA viruses are very common, not for DNA viruses, but for RNA viruses, this is very common. So remember hepatitis C virus, remember dengue virus. So quasi-species that we explained 2 weeks ago also appear during HIV infection because of these mutations, because of these mistakes during replication. A lot. Every DNA molecule that is made, between 1 to 10 mutations appear. So many different viral quasi-species infect the same patient. So HIV-1 can change very quickly, This is a problem for the immune system because the immune system cannot recognize the antigens. And this is also a problem for the antiviral drugs that we use because the target of the antiviral drugs change. So until now, this is microbiology. This is everything I have to tell you about microbiology. However, we have to say something about clinical medicine because if we don't explain microbiology inside a clinical context, microbiology is useless for you. So we have to explain a little bit clinical medicine, okay? So we are going to explain an introduction of HIV infection. HIV infection is very complicated. You need many years to study HIV to understand. So this is only an introduction. This is only a very simple introduction for medical students, okay? So what happens after exposure? What happens? So we are going to study the infectious process since the beginning, from exposure to disease, okay? As we do always. So what happens after exposure? We are going to explain this with another table. We are going to explain the natural history of HIV infection from exposure to disease. So we are going to explain the clinical phases, the clinical stages of HIV infection. What happens after exposure? So between 2 to 6 weeks after exposure, remember, exposure, sexual, parenteral, mother to child. Within 2 to 6 weeks after exposure, the patient will suffer the acute infection. This is the first stage of HIV infection, acute infection. So these patients will show non-specific symptoms, like this disease is like a mononucleosis that you already know. It's a mononucleosis-like, or like an influenza-like. So similar to mononucleosis, similar to influenza. So these patients complain about fever. These patients complain about nausea or diarrhea, even sometimes, or headache, or malaise. So they feel terrible. Or swollen lymph nodes. So as you can see, these symptoms are non-specific. So this is the typical acute HIV infection. However, this is not severe, and many patients don't notice anything. This is very mild. This is very mild because the immune system starts to fight HIV. So this acute infection resolves, cures spontaneously within 1 to 2 months. Resolves spontaneously. However, the infection does not disappear because HIV infection is a persistent infection. Persistent, chronic. Remember the meaning of chronic. Chronic infection means the virus is replicating slowly. The virus does not disappear. Chronic infection. Persistent chronic infection, mostly inside the lymph nodes. Because the lymph nodes contain T cells and macrophages. So, during this persistent chronic infection inside the lymph nodes, the virus is replicating slowly, and the virus is killing T helper cells. So there is a very slow killing of T helper cells. This is the problem. So this is the first period. The first period lasts just 1 to 2 months. So what happens later? What happens later? The second period of HIV infection is a clinically asymptomatic phase. So patients recover. Patients are asymptomatic. Patients do not show any symptom. In some papers, in some books, this stage is called clinical latency. Take care with this word. Take care with latency. What's the meaning of clinical latency? Clinical latency means the patient is asymptomatic. That's the meaning. Take care with this, because HIV infection is not a latent infection. HIV infection is a chronic infection. The virus is not inactive. The virus is not sleeping. The virus is active. The virus is replicating. So take care with this word, okay? Latency. It's clinical latency, asymptomatic. So patients have no symptoms. They can transmit the disease. Sorry, they can transmit the infection, but they don't have symptoms, okay? However, during this period, asymptomatic, as I told you before, there is a slow killing, a slow destruction of T helper cells. And this is the problem. Slowly. How much slowly? It depends on the patient. So depending on how slowly these T helper cells are destroyed, we can differentiate three types of patients. Most patients, we call them slow progressors. And what is this? These are around 70 to 80% of patients, so most of them. In these patients, they will develop AIDS after 8 to 10 years. So in these patients, the disease progresses very slowly. 8 to 10 years of asymptomatic disease. After 8 to 10 years, they will develop AIDS. These are the most common patients. However, some patients are called rapid progressors. These patients are not many, between 10 to 15% of patients. They are very unlucky. They will develop AIDS after 2 to 3 years. And finally, we have another group of patients, very lucky, that we call long-term non-progressors. They are very few. They are around or less than 5% of patients. These patients do not develop AIDS. Their immune system controls the disease, or they develop AIDS very, very late. These patients are very interesting. We are studying these patients very carefully, because if we understand why these patients do not develop AIDS, we can understand how to cure the disease. These patients do not develop AIDS, or they develop very, very slowly. However, most patients are here. So what happens when the number of T cells is reduced very much? The third stage. The third stage is called acquired immunodeficiency syndrome, AIDS. So what is AIDS? These patients either have very low number of T helper cells, so the T helper cells, so the T cells CD4 positive is the same. The T cells CD4 positive have decreased very much. to less than 200 per microliter. The number of T cells is less than 200 per microliter. And these patients with AIDS have a very low number of T cells and or they develop the typical opportunistic diseases that define AIDS. So this is the definition of AIDS. So what is AIDS? HIV positive people who develop either typical opportunistic diseases that define AIDS and or the number of T helper cells is lower than 200 per microliter. This is the definition of AIDS. This is much more complicated. In this table, I only show you an introduction in very simple words for students to understand. But understanding HIV is much more difficult. Hopefully, you will understand in the future in internal medicine. So we go back to the presentation. This is the same. So look, this is the natural history of HIV. From exposure to death. So what happens after exposure? This is the day 0 after exposure. What happens after exposure? Viremia increases. In green color, I show you here viremia. So the number of viruses in blood. So viremia increases very much after exposure. This is the period of acute infection. When viremia increases very much at the beginning, the number of T helper cells decreases because they are destroyed. In blue color here, I show you the number of T helper cells. So in the beginning, during acute infection, the number of T helper cells decreases a lot. From 1,000 per microliter to about 500. So they decrease a lot. This is the period of acute infection that lasts around 1 to 2 months. So what happens after 2 months? What happens? The immune system can control the infection. So viremia decreases very much, but not completely. The disease becomes chronic. So viremia decreases, but not completely. The virus continues replicating, but slowly. So the new stage begins. The clinically asymptomatic stage starts here. So during this stage, the virus is replicating. And what happens to the number of T cells? The number of T cells recovers a little, but not completely, but enough. The number of T cells recovers. So what happens during this period of clinically asymptomatic stage? What happens during this period? During this period, remember, this period is very long. This is about 8 to 10 years. During this period, viremia increases slowly, and the number of T cells decreases slowly. This is the problem of the clinically asymptomatic period. So what happens after 10 years? After 10 years, the number of T cells is very low. So look, approximately, when the number of T cells is below 200 per microliter, many opportunistic diseases may appear, and viremia increases very much. So this is the third stage of the disease. This is AIDS. This is the acquired immunodeficiency syndrome. The number of T cells is very low and viremia increases a lot. The patient will suffer many opportunistic diseases and the patient will finally die after a few years. This is the picture of the natural history of HIV infection. So look, this is the classification of HIV. This is not important for you now because you will study in detail next year. Just to tell you, the CDC, the Center for Disease Control, published some years ago the last classification of HIV infection. And you will learn that HIV is classified into 3 stages. HIV infection is classified into 3 stages. And the last one, stage 3, well, this classification is made depending on the number of T helper cells per microliter. When the number of T cells per microliter is less than 200, we say that the patient is suffering AIDS. So the definition of AIDS is stage 3, so T helper cells lower than 200, and or the patient suffers the typical opportunistic diseases that define AIDS. The typical opportunistic diseases that define AIDS are a lot, around 25 or 26. If you want to know the complete list of typical opportunistic diseases that define AIDS, you can go to the complement for a better understanding at the end of the class, and I show you all. Just remember, some of these typical opportunistic diseases that define AIDS are Kaposi sarcoma, that you already know, Pneumocystis jirovecii pneumonia, that you will study next year, because this is a fungus. Cytomegalovirus, that you already know. Mycobacterium tuberculosis, that you already know. Progressive multifocal leukoencephalopathy, that I told you, but I'm sure that you forgot. This disease is produced by the JC virus. It's one of the polyoma viruses that we couldn't study because we didn't have time. So remember, please, Kaposi sarcoma, remember Pneumocystis jirovecii, remember cytomegalovirus, and remember tuberculosis, because they are very common. Opportunistic diseases are not only infectious diseases. Remember, the functions of the immune system are to fight microorganisms, but also to fight cancers. So, opportunistic diseases in HIV positive people may be either infections or cancers. Infections may be primary infections or reactivations of latent infections. Reactivations are very important. Remember, tuberculosis, latent infection. Cytomegalovirus is extremely important for HIV. Reactivation. Many different reactivations. Tuberculosis, cytomegalovirus, Epstein-Barr, roseolovairuses, a lot, many different. And what about cancers? What about neoplasias, cancers? The most common are Kaposi sarcoma, you know, lymphomas, that you will study in histopathology next year, lymphomas. For example, lymphomas in the brain, non-Hodgkin's lymphoma, and cervical cancer in women related with papillomaviruses that you already know. Microbiology again. This is, again, microbiology. How can we make the diagnosis of HIV infection? The diagnosis of HIV infection is performed at the laboratory of microbiology. And we can detect antibodies by serology. We can detect viral antigens, and we can detect the RNA. So the same as for other viruses. We are going to explain this in more detail because this is important for HIV. So we have another table. So how can we make the laboratory diagnosis of HIV infection? We can detect 4 different markers at the laboratory. The first one is the RNA, the nucleic acid, so the HIV RNA. We can detect the RNA of the virus by PCR, by real-time PCR. Quantitative is real-time, quantitative reverse transcription PCR that you already know from your class of molecular biology. This is the first marker that we can detect. Second one, the protein 24. This protein, this antigen, belongs to the viral capsid. Third marker, IgM antibodies. IgM antibodies are the first antibodies that appear. And finally, IgG antibodies that appear later. So these are the 4 markers that we detect at the laboratory of microbiology to make the diagnosis of HIV infection. The first marker that appears in blood, because we detect these markers in blood, the first marker that appears is the HIV RNA. These markers appear in blood around 10 days after exposure. So this marker appears very early. This is the first one. The protein 24 from the capsid appears after around 2 to 3 weeks. So 2 to 3 weeks after exposure. The IgM appears after 3 weeks. So around 3 weeks after exposure. And disappears later. And the last marker that appears is the IgG, that appears after 4 weeks. So after 1 month. 1 month after exposure. So the earliest marker is the HIV, and the last one is the IgG. At the beginning, 40 years ago, we could only detect IgG. We didn't have immunological techniques to detect the other markers. So at the beginning, the diagnosis of HIV was possible 1 month after exposure. Currently, we have immunological techniques to detect the P24 antigen and IgM. So currently, we can detect the HIV infection very early. So the P24 antigen can be detected in the last 10 years, and this can be detected by the most modern immunological methods. They are called 4th generation, so the most modern immunological methods, immunoassays, immunological techniques, the most modern. The IgM can be detected also by the 3rd generation immunological assays. Well, obviously the 3rd and the 4th, because they are the most modern. And the IgG can be detected by all the immunological assays that we have. So by the 1st, the 2nd, the 3rd, and the 4th immunological assays. This is not important for you. This is only to show you that at the beginning, the first immunological techniques that we had for the diagnosis could detect only IgG. Currently, the most modern, so the 4th generation immunological assays, can detect IgG, IgM, and proteins from the capsid. So currently, the diagnosis of HIV infection is performed by 4th generation immunological assays. This is what I explain you here in this picture. So look, this is the same. This is exposure, the day 0. After exposure, the first marker that appears in blood is the RNA. 10 days later. The second marker is the P24 protein from the capsid that appears after 2 weeks and disappears later. And this, after 3 weeks, so 3 weeks after exposure, antibodies. These antibodies are at the beginning IgM that disappears later. And after 1 month, IgG appears. And IgG remains positive forever. So currently, as I told you, the laboratory diagnosis of HIV is performed by a 4th generation immunoassay. And finally, how can we prevent HIV infection? We are going to talk a lot about prevention. What about the basics of HIV prevention? How to prevent transmission? The prevention of transmission is very easy because we already know how HIV is spread. So the most common route of transmission in the world is sexual transmission. So how to prevent sexual transmission? By safe sexual practices, safe sexual relations. So the same as always. Using condoms by males correctly. So the risk is reduced. And this should be done every time you have any kind of sexual contact to reduce HIV transmission, but also to reduce the transmission of other sexually transmitted infections, gonorrhea, syphilis, hepatitis B. You know a lot already. This is very important. We have to teach this in high schools because currently sexually transmitted infections are increasing a lot in teenagers in the world, also in Vietnam, because teenagers don't know about how to prevent sexually transmitted infections correctly. This is very important. We have to improve in this. And something else related with HIV. We know that the transmission of HIV is easier if the patient is already suffering another sexually transmitted infection. So, patients with syphilis, patients with gonorrhea, patients with herpes simplex can transmit or acquire HIV more easily. How to prevent parenteral transmission? This is very easy. Never sharing needles. Only using a sterile injection equipment. This is very important. However, for patients who inject drugs, this is very difficult. You know that patients who inject drugs may be infected by hepatitis B, C, and HIV at the same time because the way of transmission is the same. And finally, how to prevent mother-to-child transmission? We know how to do it. The most important way to prevent mother-to-child transmission is to treat the pregnant woman with antivirals. So, women who are already taking HIV medicine should continue taking them during pregnancy as much as possible. We also treat the newborn with sidobudin. This is not important for you now. You will learn this in the future, okay? So you don't have to learn. Just remember, the best way to prevent mother-to-child transmission is to treat the pregnant woman with antivirals. And other methods to prevent that we are not going to study because this is not microbiology, okay? You will study in the future. So, prevention of transmission, okay? We also have post-exposure prophylaxis. Why do we need post-exposure prophylaxis? For many reasons. For example, if we suffer accidents when we are working at the hospital, when you work next year at the hospital, you will suffer many accidents. You will suffer accidents with needles, for example, and some of them will be contaminated with HIV, and you will need post-exposure prophylaxis to prevent HIV transmission. So what is post-exposure prophylaxis? Taking antivirals after you have been exposed to HIV. This prophylaxis should be started as soon as possible, ideally before 3 days. And you have to take this post-exposure prophylaxis for 1 month. I always explain the same. When you go to work to the hospital, you are going to suffer many accidents. I suffered a lot with needles, so many. When we suffer accidents in the hospital, many viruses can be transmitted through these accidents. The most important virus transmitted through accidents in the hospital is hepatitis B virus. That's the most important. So before you go to the hospital, please check that you have been vaccinated against hepatitis B. This is the most important. The second one is hepatitis C. And the third one is HIV. So post-exposure prophylaxis. More prevention. How to prevent opportunistic diseases in HIV-infected patients. How can we prevent that patients suffer opportunistic diseases? The best approach to prevent opportunistic diseases is to start antiviral therapy against HIV. So look, when the number of T cells is very low, the risk of suffering opportunistic diseases is very high. Also, when HIV viremia is very high, and also when the cytomegalovirus viremia is very high. I didn't explain you this because we didn't have time, but I want to tell you that cytomegalovirus is an immunosuppressant virus itself. I mean, patients who suffer cytomegalovirus disease, primary or reactivation, they may suffer opportunistic diseases because this virus is immunosuppressive itself. However, the most important risk factor to suffer opportunistic diseases is the low number of T cells. So what about this prevention of opportunistic diseases? When we prevent opportunistic diseases, we can talk about two types of prevention: primary prevention or secondary prevention. And what is this? Primary prevention is to prevent an opportunistic disease before it appears for the first time. We can perform primary prevention depending on the number of T cells. So for example, we have many different examples that you are going to study next year or whenever. For example, when the number of T cells is below 200 cells per microliter, patients have a very high risk of suffering pneumocystis jirovecii pneumonia. So we can give these patients antimicrobials to prevent this pneumonia. We can give cotrimoxazole, for example, or toxoplasmosis that you will study next year in parasitology. These are protozoa. When the number of T cells is below 100 per microliter, we can give these patients antimicrobials to prevent toxoplasmosis. We can give cotrimoxazole and many others. We have many examples, a lot, okay? Or remember, to prevent tuberculosis, we can give isoniazid to these patients, post-exposure prophylaxis. We have a lot, okay? So primary prevention, to prevent an opportunistic disease before it appears for the first time. Secondary prevention, to prevent the opportunistic disease, but to prevent the recurrence. So to prevent that the opportunistic disease appears for the second time. We have many examples that we are not going to study because this is clinical medicine and you will study this in the future. What about the treatment of HIV? We need many years to understand the treatment because this is very complex. We need a lot. We are not going to explain anything about the treatment because we have no time. Just to tell you that in the last 25 years, antiretroviral therapy against HIV has improved very much. It has reduced the morbidity and mortality a lot in the last 25 years. I remember when I started studying medicine in the year 1994, most patients that I met died because of HIV, most of them in very few years. However, in the year 1996, new drugs appeared, and that was a revolution in the treatment of HIV, and patients could survive for many, many years. So in the last 25 years, we have improved a lot. However, we cannot cure HIV. We can control the disease in many patients, but we cannot cure, not yet. Maybe in the future, but not yet. According to the World Health Organization, we have to treat all patients infected. So all patients infected with HIV must be treated with antivirals, all of them. Mostly patients with AIDS, or children younger than 2 years, or people with less than 350 T cells per microliter. We should treat all. If we cannot, the priorities to treat are these. But currently, the last recommendations of the World Health Organization are to treat all infected patients. And finally, we also have pre-exposure prophylaxis. But we are not going to say anything about this, just very few words, because this is very difficult to understand and there are many advantages and disadvantages, and this is public health. This is not microbiology. Just to tell you that currently in some countries, antivirals for pre-exposure prophylaxis are available, not vaccines, because we don't have any vaccines. This pre-exposure prophylaxis is performed by giving antivirals daily to prevent infection, but for very few people, for people who have a very high risk of becoming infected by HIV. I explain you here and also in the complements for better understanding. But we don't have time to explain this in detail, and this is not microbiology, so we are not going to say anything. So in this picture that we always use, I show you everything we can do with HIV infection. So look, what can we do here? What can we do here in this part of the cycle? The most important, we can treat all HIV positive patients with antivirals. This is the most important. We can also prevent opportunistic diseases with antimicrobials, primary prevention or secondary prevention. And we can also treat opportunistic diseases with antimicrobials. These are all the things that we can do here. We also have post-exposure prophylaxis. So, taking antivirals after exposure to prevent infection, ideally before 72 hours. For example, if we have an accident in the hospital, if we have a sexual relationship unprotected with an HIV-infected people. And we also have pre-exposure prophylaxis, but only for some people with very high risk of infection. And we are not going to explain this in more detail. So in this picture, we have everything we can do. And every year, for many, many times, I always finish the last lesson of HIV with the same slide. And I always try to explain the same. Currently, most people infected with HIV live in very poor countries in sub-Saharan Africa. Most of them are very poor, most of them. So most of them cannot get the proper treatment. During this course of microbiology, I have told you many times that poverty is a very important risk factor to suffer infectious diseases in the world and to die. Because many people live in very poor places with no access to health care. And this is not good. They live in places where no medical progress seems to have been made. This is a shameful situation for all. Some people live in very rich countries. Many people live in very poor countries. This is shameful for all. Hopefully in the future generations, your sons and your grandsons will do it much better. Because now the world is very unfair, very, very unfair. Most people live in very poor countries and very few people live in very rich countries. This is shameful for all. Hopefully the future will be better. Okay, so do you have any question about HIV?
Yes.
Question.
As you mentioned that we cannot cure HIV yet, but yes. Is there anyone that naturally immune to HIV?
We have two things here, We have less than 5% of patients that can control the disease. So they don't develop AIDS, or they develop AIDS very, very late. This is one. So we cannot do it. They do it by themselves, their immune system. This is one thing. Second thing, currently, we have 2 or 3 patients in the world that have been cured, but not with antivirals. They have been cured because of a bone marrow transplant. Because of a different disease. This is more complicated and you will study in the future. We have 2 patients in the world infected with HIV that suffer leukemia. The treatment of leukemia is a bone marrow transplant. And they were very lucky because they received the bone marrow transplant from people with some mutations in the co-receptors. People with mutations in the co-receptors do not develop the disease. So these patients infected with HIV receive a bone marrow transplant from people with mutations. And they were cured from HIV. But these patients in the world are only 2. Only 2. So currently we are studying the mutations of these co-receptors, because if we understand the mutations of the co-receptors, we understand how to cure the infection. But currently with antivirals, we can't cure the disease. It's impossible.
Okay, I understand now. Thank you.
More questions? Oi. Ko, do you do something? Ko? Any question?
Các bạn có câu hỏi gì không các bạn?
Không ạ.
No, no. They said that no, no.
Questions? No questions?
No. No.
We finish forever? Okay, so we finish with this class, the course of microbiology. We don't finish everything because we have to finish in the future the practices of laboratory. If we are alive in the future, because nobody knows, because the situation is changing very quickly, so we don't know what will happen in the future, okay? So we finish the lectures, so I wish you good luck. I remind you that the most important for you is to learn. That's the most important, okay? So good luck in the future and good luck with your final exams now, okay? We will have the final exam of microbiology in the future because I disagree to have a final exam online. That is the reason why we don't have the final exam online. We will have in the future. Okay, so goodbye and good luck.
Goodbye.
You want to say something? Go. Go. You want?